Welcome to the Deep Dive. We're here to unpack complex research and give you the core insights. Today, we're diving into prostate cancer, specifically the type driven by a really common genetic hiccup, the ERG transcription factor translocation. Well, it's been a big puzzle in cancer research.
It really has. Prostate cancer, PCA, is incredibly common. And here's the thing. In, maybe 40 to 50% of cases in Western countries, the engine behind the cancer is this gene fusion. It makes the ERG transcription factor go into overdrive.
Like sticking a supercharger on it.
Exactly. It's massively overexpressed.
And we've known for a long time that this ERG thing is a key trigger, right? You even see it early on in lesions that look mostly okay, called Pelomine prostatic intraepithelial neoplasia. But how that mistake actually kicks off a full-blown invasive tumor, that's been surprisingly hard to pin down.
Yeah, and a lot of that difficulty came down to how people were looking.
Yeah.
The older methods used bulk tissue analysis, basically grinding up the tumor and looking at the average molecular soup.
All right.
But the start of the cancer, that initiation event, it's likely happening in just a few really specific cells. Trying to find those using bulk methods. It's like trying to figure out who laid the first brick of a skyscraper by analyzing, the concrete mix from the 50th floor.
You lose all the important early details.
Completely. You miss the actual blueprint, the initial team.
Okay, so this deep dive is based on newer research using single-cell technology and this technique called lineage tracing, which is kind of like putting little GPS trackers on cells to see where they go and what they become. So our mission today is really threefold. We want to identify, 1, the specific cell type where ERG actually starts the fire, 2, this apparently very active intermediate cell state it creates, and 3, the specific molecular weaknesses, the vulnerabilities of that state, things we could potentially target.
Yeah, this work really shifts the perspective on how ERG-driven cancer gets going.
Okay, let's tackle that first big mystery. The cell of origin. The prostate lining, the epithelium, it has these two main layers, right? Basal down below, kind of stem-like, and luminal on top, the secretory cells. For ages, people argued, does ERG cancer start in basal or luminal cells? What did the new model show?
Well, the evidence landed pretty firmly on the basal layer, but, and this is key, only a very specific type of basal cell. They used a mouse model where they could switch on ERG and also knock out another common gene, PTEN.
Okay.
When they took just the basal cells from these mice, turned on the cancer genes, and transplanted them, they got invasive adenocarcinoma pretty much every time. 6 out of 6 cases, in fact.
Wow. Okay. So clear results from the basal side. What about luminal?
Much different story. When they did the same with luminal cells, the transplants resulted in much smaller benign growths and far fewer ERG-positive cells within them, maybe only about 11% compared to nearly 45% in the basal-derived tumors.
So the basal cells are definitely the preferred launch pad for invasive cancer. But you said it wasn't all basal cells. This is where the single-cell stuff comes in.
Exactly. That's where it got really interesting. The real tumor-starting ability wasn't in your average basal cell. It was concentrated in this rare subgroup they called BasalLum cells.
BasalLum. Okay, what makes them special? Sounds like a mix.
It is a mix. Think of them as having a bit of an identity crisis. They look basal. They have the typical basal markers like K5, P63. They sit in the right spot near the basement membrane. But crucially, they also abnormally express genes typically only found in luminal cells, things like TMPRSS2 and KX31.
Ah, I see. So they're sort of primed. They have the basal regenerative capacity, but already have some luminal programs switched on.
That seems to be the idea. This mixed identity signifies plasticity, a flexibility that ERG seems to exploit. It's like ERG needs that specific kind of pliable clay to really mold into invasive cancer.
And this isn't just a quirk of the mouse model.
Nope. They checked in humans. Using single-cell RNA sequencing on human prostate cancer samples, they found these rare ERG-positive BasalLum cells existing within the basal cell groups in actual patient tumors. So the mouse model seems to reflect the human situation here.
Okay, and they really nailed this context idea using different genetic switches, the Cree drivers, right? Targeting BasalLum versus just luminal.
That's right. They used a driver, NK-by31 Cree2, which targets both luminal cells and this specific BasalLum subset, when they activated ERG using that driver.
Let me guess, invasive cancer.
Bingo. Rapid expansion of ERG-positive cells within a month, and all the mice developed invasive adenocarcinoma by about 9 months.
And if they used a driver that only hit the mature luminal cells, like K8 Cree2.
Then you only got hyperplasia, maybe pin those early non-invasive changes. but never invasive cancer, even after a year or more.
So ERG is like a powerful engine, but it absolutely needs the specific context, the high-octane fuel of the BasalLum cell, to actually start the race towards invasive disease.
And to put it better myself, that context is everything for initiation.
Okay, so takeaway one. ERG cancer starts in these rare plastic BasalLum cells. Got it. All right, we've found the starting block. But the end result, the invasive cancer we see under the microscope, it looks almost entirely luminal. It forms glands, the cells look luminal. If it starts in a BasalLum cell, how does it ditch the basal features and become this massive luminal-like cells?
That's the next key piece, the transition. It doesn't just flip a switch. It goes through this intermediate phase, a highly dynamic, very proliferative state they call the intermediate or IM cell state.
Okay, an intermediate step.
Exactly.
Yeah.
Think of the BasalLum cell as writing the initial messy draft. The IM cell is like the high-speed printing press, churning out copies that are closer to the final version.
Tell me more about this IM printing press. What defines it?
Well, these IM cells are fascinating. They actually co-express both basal markers like K5 and luminal markers like K8 and K18, so they're truly in between. Plus, they show features associated with stem cells or progenitor cells found in tissue regeneration, like Hillock cell markers.
So still mixed, but maybe leaning more luminal. And you said highly proliferative.
Extremely proliferative. This is their defining feature.
Yeah.
We're talking 40 to 50% of these IM cells are positive for Ki67.
Ki67 being the marker for cells actively dividing.
Right. Compare that to the bulk of the more mature-looking luminal tumor cells, where maybe 25 to 30% are Ki67 positive. These IM cells are really driving the rapid expansion early on.
Okay, that makes sense. But how do they prove that these IM cells actually become the final luminal cancer cells? They use something called an EDU pulse chase assay. Sounds technical.
It does, but the concept is actually pretty neat. Think of EDU as a special kind of ink, a fluorescent ink, that gets incorporated into DNA only when a cell is replicating its DNA to divide. So they give the mice a short pulse of this EDU ink, maybe for just 2 hours. Which cells are going to soak up the most ink?
The ones dividing the fastest, the IM cells?
Precisely.
Yeah.
Because they're so proliferative, they get preferentially labeled. Then they wait, that's the chase period, say a week. During this time, the cells continue to divide and differentiate.
And what happens to the ink after the chase?
The ink gets passed down to the daughter cells. What they saw was that after the 1-week chase, the fluorescent ink signal had largely shifted. It wasn't predominantly in the IM cells anymore, which decrease in number over time, but was now found overwhelmingly in the K8 positive, K5 negative, mature-looking luminal cancer cells.
Direct visual proof. The ink moved from the factory IM cells to the final product, luminal cells.
Exactly, it shows the flow. BasalLum gives rise to IM, and IM gives rise to the bulk luminal cancer. They even saw direct evidence of asymmetric division, where one IM cell divides into one IM and one more differentiated luminal cell.
And importantly, this IM state isn't just something seen in the lab mice. It has real-world implications, right?
Absolutely. When they looked at human ERG-positive prostate cancer data, having a stronger molecular signature of these IM cells was associated with shorter disease-free survival for patients.
So these IM cells, this factory state, they're not just an intermediate step. They seem to be prognostically important. Targeting them could be key.
That's the hope. We've found this critical bottleneck.
Okay, so recapping, starts in BasalLum, rapidly becomes this hyper-proliferative IM factory state, which then turns out the luminal cancer. Now, the crucial question. What makes that IM factory tick? What are its molecular weak points? How do we potentially shut it down?
Right. To get at that, they needed to look deeper than just which genes were turned on or off RNA. They needed to see the underlying control panel with DNA accessibility. For that, they used single-cell ATAC sequencing, SCAT-TAC-SEC.
Okay, ATAC-SEC. DNA is the cell's instruction manual. Chromatin is like how tightly folded or open the pages are. ATAC-SEC maps the open pages.
That's a great analogy. It maps the regions of the genome that are physically accessible, the spots where regulatory proteins, like transcription factors, can actually bind and do their job, like finding all the unlocked doors in the genome.
So they looked at the unlocked doors specifically in these IM cells in the ERG-driven cancer model.
Exactly. And not surprisingly, they found a unique chromatin state, very specific to these ERG-driven IM cells, and it was heavily enriched for binding sites or motifs for the ETS family of transcription factors.
Which includes ERG itself. Makes sense. ERG is there opening up its own binding sites.
Right. But ERG, powerful as it is, doesn't usually work alone. It needs partners. So they looked at what other transcription factor binding sites were selectively open only in these IM cells.
And who showed up to the party?
They found enrichment for motifs of several other key players: STAT3, NFP, and NFAT.
STAT3 is interesting. Isn't that often involved in inflammation and growth factor signaling?
Precisely. STAT3 acts like a transducer for those kinds of signals. Its presence here suggests that ERG, in the specific BasalLum context, might be hijacking or cooperating with these fundamental stress and growth pathways to drive proliferation.
Okay, so ERG opens the doors and partners like STAT3 rush in to amplify the go signal. But they didn't just look at the switches, transcription factors, they also looked at the crew that physically remodels the chromatin, right? The epigenetic regulators.
That's right. They looked at gene expression again, specifically in the IM cells, for enzymes that modify histones the protein's DNA wraps around, and two popped out with high expression: KMT2A, which you might also know as MLL1 and DOT1L.
KMT2A, MLL1 and DOT1L. And these are histone methyltransferases. What do they do?
They add methyl groups, little chemical tags onto histones. Think of them as the maintenance crew that puts sticky notes on the chromatin, essentially ensuring that those specific access doors, the ones ERG and STAT3 want open, stay open consistently. They help lock in that oncogenic state.
Wow. Okay, so this paints a picture of a whole molecular team. ERU and STAT3 are calling the shots, and KMT2, ML1, and DOT1L are physically modifying the structure to make sure those shots can be called effectively and repeatedly, a specific dependency of this IM state.
Exactly. A vulnerability that you'd likely miss completely just by looking at the bulk tumor average.
Which leads directly to the next logical step. Are these partners actually needed for the cancer to grow? Can you break the machine by removing one of these parts? So they tested that directly, using CRISPR to knock out these genes in the cancer cells.
Yes. They used tumor-derived organoids, basically mini tumors grown in a dish, and used CRISPR to delete the genes for STAT3, KMT2A, or DOT1L individually. Then they transplanted these modified organoids back into mice to see if they could still form aggressive tumors.
And the results. What happened when you took out STAT3?
Knocking out STAT3 significantly reduced tumor growth. Actually, the effect was comparable to knocking out ERG itself.
Whoa. So STAT3 is just as crucial as the main driver. What about KMT2A-MLL1?
Same story. Deleting KMT2A-MLL1 impaired tumor growth just as much as deleting ERG did.
And DOT1L.
Also the same. Deleting DOT1L severely hampered tumor growth, again, comparable to the effect of deleting ERG.
That's incredibly strong evidence. It means the aggressive cancer state isn't just driven by ERG. It fundamentally depends on this whole team. ERG, STAT3, KMT2A, and DOT1L. Taking out any one of them causes the whole thing to falter.
Precisely. And interestingly, in the few small, non-invasive bits of tumor that sometimes grew even after deletion, they saw that ERG was still expressed, but the deleted target like STAT3 or KMT2A was truly gone. It reinforces the idea that they all need to be present and active together for the full invasive program.
Now, there was an interesting detail about KMT2A-MLL1 and a protein called menin, right?
Yes, that was a fascinating nuance. See, KMT2A-MLL1 often works by binding to menin, and there are drugs in development, menin inhibitors, that target this interaction. So the researchers thought, okay, let's delete the gene from menin, MEN1, in our mouse model.
And that should have stopped the tumors if KMT2A needs menin here.
That was the expectation. But surprisingly, deleting menin did not significantly impair the ERG-driven tumor growth in this specific model.
Huh. Why is that important?
It suggests that, at least in this particular context, this highly plastic BasalLum to IM transition state, the crucial role of KMT2-MLL1 might actually be independent of menin. It's cooperating with ERG through potentially a different mechanism here.
And that matters for treatment, doesn't it?
It does. It implies that maybe menin inhibitors, while useful elsewhere, might not be the best way to target KMT2A activity in this specific subset of ERG-positive prostate cancer.
Yeah.
You might need drugs that inhibit KMT2A's enzymatic activity directly.
Right. But regardless of that specific mechanism, the really exciting part here is the translational potential. You're saying there are already drugs being tested in clinics that target STAT3, KMT2A-MLL1, and DOT1L.
Yes, exactly. Clinical-grade inhibitors exist for all three of these partners. This research essentially hands us a rationale based on the specific vulnerability of that IM cell state for testing combinations of therapies targeting these pathways in ERG-positive patients. It's moving beyond just targeting ERG itself. So to kind of wrap it all up for you, this deep dive reveals that ERG-driven prostate cancer isn't monolithic. It's really a story about context. It kicks off in these specific rare plastic basal cells. They then rapidly morph into this highly proliferative stem-like intermediate or IM state, that's the factory. And that factory, which drives the invasive luminal cancer, is critically dependent on a unique molecular team, ERG working together with STAT3, KMT2A, MLL1, and DOT1L.
And the big takeaway here, I think, is the power of looking at the single cell level. Combining that with lineage tracing allowed researchers to move way beyond just classifying the cancer broadly. They could pinpoint the exact cellular state, that IM bottleneck where the aggression is really being driven, and identify its specific molecular dependencies, vulnerabilities that were just, averaged out and lost in the noise of bulk analysis.
It really paves the way for more precise therapies, targeting the specific cellular transitions and dependencies that fuel the disease, rather than just hitting the whole tumor mass indiscriminately.
Which leads us to our final thought, something for you to mull over. We heard that this dependency on epigenetic regulators, specifically KMT2A, MLL1 and DOT1L, isn't unique to ERG and prostate cancer. It's also been seen in other cancers driven by the same family of transcription factors, the ETS family, like G-stumors driven by ETV1. So here's the question. If multiple different cancers driven by this ETS family share this fundamental reliance on KMT2A, MLL1 and DOT1L to stay aggressive, could targeting these epigenetic pathways be perhaps a more universal strategy, a way to treat a whole class of ETS-driven cancers well beyond just prostate?
That's definitely the broader implication, and it's a question researchers are actively pursuing right now. It holds significant potential.
Food for thought indeed. Thanks for joining us on the Deep Dive. We'll catch you next time.